Hey guys, it's Rachel, and I'm doing my presentation on CRISPR-Cas9. So I'm sure you guys probably know about it just because it's kind of like a really big deal nowadays, but basically CRISPR-Cas9 is a genomic editing tool. It's used to modify different parts of the DNA sequence to kind of figure some things out. So CRISPR stands for clustered regularly interspaced short palindromic repeats. And so kind of all that to say is it's a bacterial defense system that forms the basis of CRISPR-Cas9 genome editing technology. The Cas9 part of it stands for CRISPR-associated protein 9, and that's basically just an enzyme that's used to cut parts of the DNA. So I like to think of it as kind of like the molecular scissors of this whole process. So CRISPR-Cas9 enables scientists to be able, and medical researchers, to be able to edit like those different parts of the DNA by removing and adding and altering those specific sections. And this is super important to researchers because they're able to create different mutations or delete mutations for like furthering research as far as like certain diseases, so like cancer. I know that they've tried it for HIV and AIDS, and so just kind of like those diseases that have to do with like the genetics. It's like super popular for that, and like scientists are really loving it because of that. And so it's actually currently the simplest and most versatile, as well as most precise and the cheapest method of genetic manipulation or modification. I was like looking at it, and it was like for other tests that like kind of do the same thing, they were like over $1,000, and with CRISPR-Cas9, I don't know, like I didn't really look super in depth because that's not really what like I was focused on, but it was like saying that like you could get a DNA like kit and everything for $65, which is like super good because obviously when you were doing research, I mean you have money, but when you can save money, that's what you want to do. But not only is it the cheapest, like I said, it's the simplest and most precise, and it's most versatile. So like it literally, other tests can take months to figure like to get those results, as well as you can only do one at a time. And what I mean by versatile is that you can perform multiple, you can do these on multiple genomes at the same time. And get these test results back in like days to weeks. So that's just like a huge difference, doing one, getting it back in a few months versus doing a couple and getting them back in a few weeks. So obviously it's not really like super debatable about like, oh, what's the best one right now. So that's kind of where like this method has most of the other ones beat out. So it also has like a super high degree of flexibility and accuracy when copying and pasting that DNA. So that just is another thing, kind of goes into like the precision of it, as well as like how versatile it is. Just like other things like help like put it above other tests and things like that. So it has a super wide variety of applications, in terms of like you can use it for basic biological research, or like developing biotechnology products, and like I said, treatment of diseases, which is kind of like what I focused on more so. But yeah, so I think that covers everything. Oh, so there's two components which I'm actually going to cover in the next couple of slides, but the two main components are the CRISPR and the Cas9. The CRISPR is basically made up of, it codes for cRNA, which is just CRISPR RNA. And then the CRISPR-associated protein, like I said, is like the molecular scissors for this whole process. So yeah, just kind of like applications. I wanted to include this middle one because, like I said, it's also a very ethical thing right now. So obviously it has potential to be able to treat several medical conditions. Like I said, it can be used to help figure out cures for cancer, or HIV or AIDS or things like that. But this one is like probably one of the bigger things that I think it's most known for. And that's that it's involved with somatic cells, and like the editing of those or like altering of those, which is just like non-reproductive cells. So like that's all cool. But the problem that people are having and like debating over is people are wanting to include this and like involve CRISPR into germline cells, which the problem with that is that it's like with germline, you like snip out something that you don't want, and then generations after generations after generations won't have that either. So it's like, okay, well, is that playing sorry, that was a voice crack. So it's like, is that playing God by doing that? By like, okay, I don't want my son to have or my daughter to have brown eyes, so snip that. And I don't think it's really like quite that simple, but kind of like that basis of like, okay, well, I don't want them to have this or them to have this, and I want them to have this. So it's like, what part of that do you like kind of like stop or whatever? So that's, I think, what it's most known for. And kind of like my parents are very uneducated in the sciences, and they like, that was the first thing they said when I said I was doing this project, was like, oh, that's the whole thing about playing God. So I think that's probably what most people know it for. But anyway, back to the genetics of it and that thing, because that's what we really are here for. I hope anyway. Is that it's already being used to treat human diseases on a very small level. So like I keep saying, cancers and HIV and AIDS and those types of things. It's already being tested. I actually saw that it was either February or March. I guess it's April now. But within the past just couple months, they actually the past couple months, someone has been injected with CRISPR-Cas9, and that's the first time it's ever been a thing because it's like illegal to be doing in the UK and other parts. And so they're kind of in like the testing process of that on actual humans now, not just like animals or in plates and things like that. So that's kind of cool to see for like the future or whatever. So like, how does CRISPR-Cas9 work in general? So like I kind of did mine kind of twofold. So I did mine in general, but I also wanted to highlight what it kind of means in terms of results in actual studies and experiments. And so my experiment is that I kind of picked, or my study is on ovarian cancer cells, which is like super interesting to me. So we'll get into that in a little bit, but so basically CRISPR-Cas9 consists of two main components that introduce the mutation into the DNA. So you have your guide DNA, which binds to the Cas9, and the Cas9 is like this whole pink? I don't really know what color it is. This whole blob that looks like a kidney bean or a beanbag chair, whatever. And so that's your Cas9 enzyme or protein. And so your guide RNA, which is like kind of the whole CRISPR part of it, that's going to bind to your Cas9 and form that complex of the Cas9, CRISPR-Cas9. And so then what it does next is it's going to target what's called PAM sequence. Let me back up a little bit. So like the guide RNA, which I'm pretty sure we've read about, but the gRNA is what it's also called. It's a short synthetic RNA that's composed of two different things. It's composed of a scaffold sequence called TrkRNA or crRNA, which means transactivating crRNA. It's a lot of letters. And so that's necessary to be able to bind to the Cas9, where it also includes a spacer, and that defines the genomic target that's to be modified. So the spacer is kind of like what is going to bind to the PAM, or the protospacer adjacent. adjacent motifs. And so the Cas9 protein typically binds to the gRNA, and then the gRNA guides the Cas9 to that specific location marked by the PAMs. And then that's where it's cut. And so, PAMs are basically just short DNA sequences. And so, like, the way I like to remember it is that, like, they're kind of like those people at the airport with, like, the flags that are, like, waving the airplane to, like, what direction they need to be going. That's kind of like what PAMs are